Hello everyone, welcome to this biology quick review for your ATI TEAS exam. This is part 1 of 3 quick reviews that are going to be biology focused. Go ahead and grab all your snacks, grab your coffee, because we're going to go ahead and jump right in, and we're going to begin with the scientific method. The scientific method is a systematic way to find the answers to questions or problems of the natural world. It's used to explain all of the wonders of the world and also used to question the wonders of the world. You may recall that the scientific method is performed in steps. The first step is always observation. We observe something of the natural world and we form a question or a hypothesis about it. Our second step is the hypothesis. The hypothesis is an informed, logical, plausible explanation for what we observed. One important thing about the hypothesis is it must be testable. So you must be able to construct an experiment to prove that the hypothesis is correct or not. Our third step is experiment. This is where we test our hypothesis. In our experiment, we collect measurable data that we use to analyze the result and come to a conclusion. The one most important thing that you have to remember about this step, the experiment, is that you're going to have an independent variable and a dependent variable. So let me just give you a really quick example of an experiment so you can identify the independent variable, dependent variable, and also the constant variable. Okay, so let's just say, for example, here, we are conducting an experiment on a few plants. We have 3 plants here, and let's just say we want to know if feeding them a different type of liquid will change the results of the way they grow. The independent variable is the one thing that you can change in an experiment. It's the one thing that we will manipulate. And in this case, it's the type of liquid we're using on each plant. This independent variable is going to be manipulated or changed in each plant. Now, the dependent variable is the result of the independent variable. So in this case, it's what results will the plant have as an effect from the liquid that we gave them. So if we gave the first plant Coca-Cola and the plant withered up and died, then withering would be the dependent variable. That is a result of the independent variable. If the next plant we gave orange juice and let's just say its leaves got more green and bright, then our dependent variable would be the leaves becoming bright green. This is a result of the orange juice. And let's just say that the third plant we gave water to, and this plant in particular grew very, very tall. Now, the independent variable is the water, and the dependent variable is the growth, the height of the plant. The plant grew very high. Now, everything else in the experiment must be what we call controlled. Everything must remain constant and unchanged. Therefore, we know that what is causing the dependent variable or the outcome or the data, the results, is in fact the independent variable, the change in the type of liquid that we're feeding each plant. Some examples of a constant variable in this particular example could be the amount of liquid that we give each plant. So you see that's different than the type of liquid we're giving each plant, right? So the amount of liquid has to be constant. The amount of sunlight we give each plant can be a constant. The type of soil we're giving each plant can be a constant. And let's say how big each pot that we put each plant in can be a constant. So everything else, all other variables must be constant except for the independent variable, which is being manipulated. Alrighty, so as far as the experiment portion of the scientific method goes, I personally feel it's the most important. You must know the difference between independent variable and dependent variable. And if somebody gives you a question, you must be able to if someone gives you an experiment, you must be able to point out the independent and dependent variables in the experiment. The final step of the scientific method is the conclusion. In the conclusion, there are 3 important things that you must include. The first is the summary of the overall experiment. The second is the results of the experiment. And the third is whether you accept or reject the hypothesis. Next, we're going to jump into the biological hierarchy. This is just a systematic way of putting every living thing into levels of complexity. So we begin first with the atom. The atom is what makes up all matter. It also makes up molecules. Molecules are any 2 or more atoms put together by a chemical bond. Molecules are what make up cells. Cells are the basic building blocks of all living things. Cells are what make up our tissues, and our tissue is a group of cells that have similar structure and that function together as a unit. Tissues are what make up our organs, and an organ is a collection of different types of tissue that structurally form a functional unit specialized to perform a specific function. Our organs create our organ systems. One example of an organ system is the cardiovascular system, and its main organ is the heart, which helps pump blood, but it also includes the accessories to the heart that help the heart pump blood and take it to where it needs to go. So it includes all of the blood vessels that is very crucial that work hand in hand with the heart to spread blood throughout the body. Now, our organ systems work together to create an organism. An organism is an individual, animal, plant, or single-celled life form. So any living thing, any singular living thing is an organism. And any organism is going to have all of the things we just discussed included in it. So it's going to have organ systems, organs, tissue, cells, molecules, atoms, etc. After organism, we have population. When you have a group of organisms of the same species grouped together, we call this a population. So for example, we have a group of humans, the human species, and they're grouped together. So this is one population. In this other photo, I have a group of bats hanging out together, and they are of the same species. And they are multiple organisms hanging out together. So we call this a population. Now, populations build communities. And a community is a collection of different types of populations of species living together. So a lot of people get population and community mixed up. So the major difference is that population is referring to one particular species living together, and community is referring to different populations of different species living together. So there's a population of these bats, and they actually live in this community in this cave in New Zealand. The reason why this cave is a community is because there are different types of species living in it. There is the population of bats, but there's also this population of cave fish that you see this guy holding. And there's also this population of this really gnarly fungus that is glow in the dark that you can see here. We have these 3 different types of population, these 3 population of 3 different species living together. That makes this a community. So after we have community, we have an ecosystem. An ecosystem is all of the communities in a particular area, but it also includes the physical environment in which they live in. Ecosystem basically includes the community and population and all the organisms, but added to it is the physical environment, like the trees, the ocean, the grasslands, the mountains, everything in which they live in that is part of Earth, that is an ecosystem. Next, we have a biome. A biome is a really, really large area, environmental area that has very unique characteristics to that area. It shares very similar types of temperature, soil type, maybe water type, amount of light, similar type of bacteria, flora, fungi, fauna, things like this. Very similar types of species like animals that occupy this area. So our biomes are a collection of all the different ecosystems, but they share very similar climate conditions or environmental conditions in general. So here I have a picture of some of our most common biomes. We have grassland, savanna, desert, marine, freshwater, tundra. All of these have very distinct, let's just say, temperature ranges, soil type, species, and the picture kind of makes it very easy to see the drastic differences. So moving on, lastly, we have our biosphere. The biosphere includes all of the biomes within it, and it includes all the places on Earth where life exists. And this, my friend, completes the biological hierarchy. Alrighty guys, moving on to the pH scale. Although pH scale is super simple and there's not much you need to memorize about it, it's still very important in biology, and it's one of those high yield questions that you might get on your TEAS exam. So if you know the basics of the pH scale, you can go ahead and skip ahead, but if you need a refresher, stick around. This will only take a minute. First thing first, the pH scale is the measurement of hydrogen ion concentration in a water-based substance. So your acids are going to have a high concentration of hydrogen ions, but your bases are going to have low concentrations of hydrogen ions. So you have your acidic on one side and your alkaline or base on the other side. And you have your neutral smack in the middle. Your neutral is number 7 and only number 7. Every other number belongs to either an alkaline or an acidic. So our acidic solutions are going to be numbered 1 to 6, and our alkaline solutions are going to be numbered 8 to 14. So here are some very common solutions that you will find on our pH scale. Remember that number 7 is your neutral. The further you go to your right, closer to 14, the more alkaline or basic a substance gets. And the further downwards you go to 0, the more acidic solution gets. Alrighty everyone, so we're going to move on to macromolecules, or what it's also called, the organic molecules. Now, there are 4 macromolecules: proteins, carbohydrates, lipids, and nucleic acids. As far as the ATI TEAS exam goes, there's really only 3 things you must know about each of these. 1 is what they're composed of or made of. 2 is the structure of the molecule, and 3 is their functions in our body. The 4 macronutrients that we're going to be studying are the carbohydrates, lipids, proteins, and nucleic acids. Macronutrients are nutrients required in large amounts that provide energy needed to maintain bodily functions and carry out the activities of daily life. Macromolecules are associated with this process called hydrolysis. Hydrolysis means that when the molecules enter water, when they're immersed in water, they can be easily broken down into smaller molecules. So essentially, we have these macromolecules like fats, carbohydrates, and proteins. And when they enter our digestive system, the water breaks them down, also with the help of enzymes, breaks them down so that those nutrients or macromolecules are easily absorbed into the bloodstream. So just remember that word hydrolysis. The true definition of it is the chemical breakdown of a compound due to reaction with water. So just remember that in hydrolysis process, compounds are immersed in water and enzymes help break down these compounds into smaller substances. So let's first start with what these macromolecules are made out of. They're made out of something called monomers, and monomers are just building blocks of smaller molecules that build big molecules. Carbohydrates are made out of a monomer called monosaccharides. Lipids are made out of fatty acids and glycerol. Proteins are made out of amino acids. And nucleic acids are made out of nucleotides. All of these are monomers that help build our macromolecules. So how are these 4 macromolecules used in our body? Starting with the carbohydrates. Carbohydrates are used as a fuel for our body. Your body breaks down carbohydrates into glucose, and glucose is the main source of energy for your body's cells, tissue, and organs. There are 3 types of carbohydrates. There's your monosaccharides, disaccharides, and polysaccharides. Remember that mono is referring to one or singular. So monosaccharides are your singular simple sugar. Di means 2. So disaccharides are 2 monosaccharides that are bonded together. Poly means many. So polysaccharide is referring to a long chain of multiple or many monosaccharides. Common monosaccharides are glucose and fructose. Common disaccharides are sucrose and lactose. And common polysaccharides are starch, cellulose, and glycogen. So next we have our lipids, and remember that the monomer of the lipids is fatty acids and glycerol. These are the building blocks of lipids. It's what make up the lipids. So we're going to go into a lot more detail with the lipids because there's a lot to know here. So hang in there, guys. If you're not already bored, you might just get bored here. So just hang in there. So first and foremost, when we talk about lipids, we're talking about fats. And there are 3 types of lipids. There are your triglycerides, your phospholipids, and your steroids. Triglycerides include your animal fats and your oils. There are significant differences between these 2. Animal fats are normally solid at room temperature, and oils like vegetable oil, avocado oil, olive oil, these are liquid at room temperature. And the reason why animal fats become solid and oils are kept as liquid has to do with their molecular structure. So your animal fats are what we call saturated fats, and your oils are what we call unsaturated fats. On a molecular level, this is what it's going to look like. Saturated fats is going to have generally a carbon-hydrogen atom chain, and all of your carbon atoms are going to be connected or bonded by a single bond. So single bond is represented by this single line. So that's the takeaway here, is that your saturated fats, your carbons are going to be bonded by a single bond. There's going to be single lines between them. Now, your unsaturated fats, they look a little bit different because they have at least one double bond between their carbons. So it might look a little bit like this. You have carbons with single lines, but maybe you have 1 or 2 double bonded carbons linked to each other. This is going to make it a unsaturated fat. And in fact, these double bonds is what changes the melting point of the substance. So this is what makes these oils liquid at room temperature, basically. So this is generally what you need to know. Triglycerides, again, you have your animal fats and your oils. Your animal fats are saturated fats, meaning molecularly the carbon to carbon bonding is going to be singular. And then your oils are unsaturated fats, meaning on a molecular level, your carbon to carbon, you're going to have at least one double bond. And also remember that saturated fats are solid at room temperature, like your animal meats, and your unsaturated fats are liquid at room temperature. Let's go ahead and move on to our second type of lipid, the phospholipid. So the major thing about a phospholipid is that it has 2 sides to it. It has this head that is made out of glycerol, and remember that glycerol is one of the monomers that help build and construct the lipid. And attached to this glycerol is a phosphate group. Now, this phosphate group, molecularly, it would look something like this, but what's really important about this phosphate group is that it makes this This part of the phospholipid, it makes it hydrophilic, meaning hydro meaning water, philic meaning loving. So this part of the phospholipid is water loving. And the other side of the phospholipid are these two little legs that come out here, and these are made out of fatty acids. Now, these fatty acids are, these are called the tails. My bad, not the legs, the tails. So these tails are made out of fatty acids, and they are actually hydrophobic, meaning they hate the water. So to sum that up, phospholipids have a hydrophobic side and a hydrophilic side. Your phospholipids form a bilayer, and this is called or referred to as a lipid bilayer or a phospholipid bilayer. Now, your outer part of the layer is going to be the head or the hydrophilic side of this lipid. This is the water-loving side. And on the inside of the layer is going to be your hydrophobic or your water-hating side. It's going to be safely on the interior. These phospholipid bilayers are super important because they make up our cell membranes. They are a protective layer of our cell membranes, specifically because they have the hydrophobic and hydrophilic side. So for most organisms and most viruses, this is going to make up a major part of your cells. So the final lipid that we're going to learn about are our steroids. A steroid is any compound with a specific 4-ring structure. Now, steroids apply to various types of chemicals. One important steroid is cholesterol. Cholesterol is important because it is a precursor to the synthesis of other steroids such as testosterone, estrogen, vitamin D, and cortisol. Another common steroid is aldosterone. Aldosterone maintains fluid balance in your body by promoting reabsorption of excess sodium and excreting potassium. The last steroid I want to touch on is cortisol. Cortisol is important because it has anti-inflammatory effects on the body. Related to cortisone is a classification of drugs, man-made drugs, called corticosteroids. These are a type of anti-inflammatory drugs, just like cortisone, anti-inflammatory. These are given to patients for treatment like rheumatoid, arthritis, lupus, vasculitis, things like this that have that the cause is too much inflammation in the body. So in my opinion, as far as the ATI TEAS exam goes, there's not too much you need to know about steroids. The most important thing is that what is unique to them is that 4-ring structure. So if you see the 4-ring structure, you're going to need to identify it as a steroid, but also know some of the high yield types of steroids. From my research through various books that have review questions, I've seen aldosterone, cortisol, vitamin D, testosterone, and estrogen. And I think testosterone, estrogen, and what's the other one? Progesterone. Progesterone is also one of those high yield steroids. So guys, I am going to cut this short right here. I know that we still have to finish our macromolecules. We went over carbohydrates, we went over lipids, but we still need to go over the proteins and the nucleic acids. I'm going to stop right here because I am actually feeling a little sick and I have a feeling that I'm going to be out for the next couple days. So I will upload part 2. And the first thing that we'll jump into is the proteins and then the nucleic acids, and then we'll continue from there. But hope you enjoyed this video. If you are still watching this video to the end, please give me a thumbs up, and happy study, guys.